and welcome to our JASN podcast. Alan Yu from the University of Kansas Medical Center, Basic Science Deputy Editor for JASN. And I'm joined today by Dr. Morgan Gallazzini and Eloïse Marques to discuss their new paper in JASN, which is entitled CLUH Inhibition by Lipocalin-2 Orchestrates Mitochondrial Disruption and Contributes to Kidney Disease. So, Drs. Gallazzini and Marques, thank you for joining us. Perhaps we could start by asking you to briefly introduce yourselves and your background.
I will start. So I'm Morgan Gallazzini, and I really would like to thank you for inviting us to participate to this podcast for JASN and to give us the opportunity to talk about the study we just published in JASN. And so I'm a researcher. I'm working in a center on research on inflammation at the Hospital Bichat in Paris, where I'm leading a group that is interested in the role of cellular stress response in the development of chronic kidney disease, and with a special focus on the lipocalin protein that we will talk about. My formation is a physiology and a pathophysiology researcher.
And hi everyone, so I'm Eloïse Marques. I'm currently a research associate at the MRC Mitochondrial Biology Unit in the University of Cambridge in the United Kingdom. But the work we will talk about today was done during my PhD with the supervision of Dr. Morgan Gallazzini here today. The focus of my PhD was to understand how lipocalin-2 was inducing mitochondrial dysfunction, especially in a context of chronic kidney disease.
Fabulous. Thank you so much. So just to start off and place this in context, I think many of the listeners will have heard of lipocalin-2, perhaps for clinicians better known as NGAL or neutrophil gelatinase associated lipocalin. And stop me if I'm misspeaking about this. I was looking back, and this really came on the scenes in nephrology in about 2003, and one of the seminal papers was actually published in JASN by Prasad Devarajan, who showed that NGAL was one of the most upregulated genes early on after ischemia reperfusion injury. And we now know this as a well-known biomarker of acute kidney injury, of tubular damage, and even of CKD. And I was looking this up, and apparently there is now actually an FDA-cleared test of this that's currently approved for use to assess AKI risk in pediatric patients in the ICU. So the obvious question that I'm sure many people have wondered about is, well, if it's so upregulated in kidney injury, what's it doing, right? Is it involved in driving the pathogenesis of kidney injury, or is it potentially protective? And does it have different roles in acute kidney injury and chronic kidney disease? Perhaps you could tell us a little bit about what's already known about lipocalin-2 in kidney disease. I know it's a complicated area.
Yeah. You're right, Alan. Actually, it's a very interesting point. As you say, lipocalin-2 is well known in the field of nephrology because it's a neutrophil gelatinase associated protein, the NGAL protein, that is a small secreted protein that was first characterized actually in innate immune as an innate immune protein because it has this ability to bind the siderophores and regulate the iron handling. And it was originally studied in the context of bacterial infection and inflammation. But in nephrology, lipocalin-2 gain intention because it's one of the earliest and the most sensitive biomarker of the acute kidney injury. In fact, it rises in urine and the plasma within hours after tubular damage induced by injury, long before actually the creatinine increases, which make it actually extremely valuable clinically speaking, as you said. That there's even studies, clinical studies that are going on about that. But over time, it became actually quite clear that lipocalin is just not only a biomarker, it's actively involved in the disease mechanism. In acute settings, for instance, it's participating to the stress response, it's modulating the iron availability, and it can influence cellular mechanisms such as autophagy, cell survival, and proliferation. However, in chronic kidney disease, persistent lipocalin expression correlates with the decline of the GFR, the installation of the fibrosis, and the tubular atrophy, and the onset of the disease. So it's very interesting, and mechanistically, previous work, including ours, showed that lipocalin is able to activate multiple pathological pathways such as the EGFR signaling, the STAT3 activation, ER stress response, so on and so forth. And it was demonstrated with our latest work that it can even induce mitochondrial dysfunction such as fragmentation and reduce the mitochondrial mass and function here. So before our study, we knew that lipocalin was at the intersection between inflammation, stress signaling, metabolic dysfunction. But we didn't fully understand how lipocalin, mechanistically speaking, was connected to the mitochondrial homeostasis. So that's where we went with our study.
Thanks, Morgan. And is my understanding correct that lipocalin-2 has both protective and potentially deleterious roles? And does it depend on whether it was, or was it previously shown that it depended on whether it was AKI or CKD?
Really reflect a broader biological principle between the context and the duration that define the function here of lipocalin and the mechanistic that is important in the lesions. In fact, in acute kidney injury, lipocalin is strongly and rapidly induced and is part of the adaptive stress response that's taking place. And this short-term elevation of lipocalin expression will help the tubular cells, for instance, to cope with injury, for example, for limiting the iron-driven oxidative damage or activating stress pathway and hence coordinating the repair responses. And as you said at the beginning, some studies even suggest that actually the lipocalin has a protective role in AKI model. But in chronic kidney disease, the situation is very different. Instead of having a transient spike of lipocalin expression, this expression remains persistently elevated, and the chronic activation of the stress pathways tends to become maladaptive. So the prolonged lipocalin signaling can continuously activate, as I said, the EGFR pathway, the stress response, and that, as we will show now, will even inhibit mitochondrial function by regulating CLUH expression here. So it's in fact, it's not that lipocalin change its role between the AKI and the CKD. It's that acute or tightly regulated stress response can be either adaptive, whereas chronic unresolved signaling drives the pathology.
Perfect, yes. And I've been reading a lot of AKI to CKD transition papers, and maladaptive responses seem to be the general theme, and this seems to fit right in with it. So in terms of trying to address the mechanism, this is 2026 or 2025, I guess, when you did this. And so taking an omics and unbiased approach is really the way to go. And so it seems like you decided that the way to understand this was to figure out what are the proteins that interact with lipocalin-2. So can you talk about how you determined this interactome and just very briefly describe the key findings?
Yes, so basically we wanted to understand why lipocalin-2 was behaving so differently in acute versus chronic kidney disease. And we realized that we needed to know a little bit more about which protein lipocalin-2 was interacting with within the cells. And surprisingly, despite how it was important, lipocalin-2 interactome had remained lately uncharacterized. So we decided to address these questions, and for that we decided to use a technique that we call BioID, which allows us to identify the different protein that comes in close proximity or are interacting with the protein of interest. So for that, we engineered a fusion protein in which lipocholine-2 was linked to a biotin ligase, and we created a construct that is called lipocholine-2 BioID. We transfected this fusion protein in the cells, and then we added biotin in the media, which labeled all the proteins that were in close proximity or interacting with lipocholine-2. We then decided to purify the biotin-tagged proteins and analyze them using mass spectrometry. And this approach gave us an unbiased and comprehensive view of the lipocholine-2 interactome. And with this, we found that lipocholine-2 was interacting with a lot of different proteins involved in different cellular processes, such as Morgan said, the stress responses or cell cycle, DNA repair, and those processes that we and others described in the past literature. And many of those pathways are also directly linked to apoptosis, proliferation, or even fibrosis, which are key central features of kidney disease progression. We also confirmed previous data with the effect on mTORC1 and mitochondrial proteins. And most importantly, we identified CLUH as being a strong interactor of lipocholine-2. And this protein was really, really interesting because CLUH is known to regulate mitochondrial homeostasis, suggesting a direct link between lipocholine-2 and also mitochondria.
Thanks, Eloïse. Terrific. So CLUH is CLUH, I guess. Is that?
Yes.
I love that little nickname. So can you give us maybe a high-level overview of the mechanism that you figured out for how lipocalin-2 regulates CLUH and what its downstream targets are?
Yes. So after identifying that CLUH was a major interactor of lipocholine-2, we wanted to understand how this interaction was affecting CLUH's function and all its downstream targets. So first, we wanted to confirm that there was a physical interaction between CLUH and lipocholine-2, and we've done that using co-immunoprecipitation assay. To note, CLUH is a RNA binding protein that will bind different mRNAs encoding nuclear encoded mitochondrial proteins. And this will support their translation and stability. And this will also play a crucial role in mitochondrial homeostasis and also the distribution of mitochondria within the cytoplasm. Given our previous work showing that lipocholine-2 was regulating mitochondrial dynamics and function, we hypothesized that lipocholine-2 might influence mitochondria through its interaction with CLUH. So we examined CLUH's expression, and we found out that lipocholine-2 expression was not changing CLUH mRNA or protein levels, but we observed that lipocholine-2 expression reduced the protein levels of several CLUH targets genes, such as ATP5A, GOT2, OPA1, but without affecting the mRNA levels. This was indicating that lipocholine-2 was inhibiting the translation of those target genes. Then to understand the mechanism, we performed a RNA immunoprecipitation experiment, and we found out that lipocholine-2 was interfering with CLUH's ability to bind its target mRNAs. So lipocholine-2 was binding directly CLUH to inhibit its RNA binding activity, and this was causing mitochondria to cluster around the nucleus and also to become shorter. We also tested whether this effect was mediated through mTOR pathway, since both lipocholine-2 and CLUH have been linked to the mTOR signaling by we and other people. And however, we did confirm that lipocholine-2 activated mTOR independently of CLUH, and indicating that this regulatory pathway and mechanism was separate from the one we see from lipocholine-2 on CLUH. And although we do not know the precise molecular mechanism by which lipocholine-2 is regulating CLUH, our data indicates that this regulation involves the cytosolic form of lipocholine-2 rather than its secreted form. So altogether, our data suggests that lipocalin-2 binds CLUH directly and acts as a negative regulator of RNA binding function. By blocking CLUH's ability to support translation of the different target's gene, mitochondrial genes, lipocalin-2 disrupts mitochondrial protein synthesis, alter mitochondrial organization, and then will contribute to mitochondrial dysfunction.
Thank you so much, Eloise. That's really clear. So these were studies done presumably all in vitro, right? And so you have to test at least some of this and show in mice that this works. So can you just briefly take us through what you did there?
Yeah, so as you say, our hypothesis was that lipocalin was disrupting the mitochondrial homeostasis by inhibiting the CLUH activity. And we first had to confirm that this correlation between lipocalin expression and loss of activity of CLUH was true in vivo models. So we used different CKD-induced mice models, such as subtotal nephrectomy surgery models or ADPKD model by the knockout of PKD1 to monitor the expression of CLUH and characterize actually the expression of CLUH in the kidney that was not described before. And we observed that CLUH is highly expressed in the nephron cells, in the tubular cells all along the tubule, and that upon injury, CLUH expression is strongly decreased while lipocalin expression is strongly increased, and that targets that were defined before for CLUH were down-regulated in expression when CLUH was down-regulated. So we confirmed that CLUH down-regulation was correlating with the onset of the lesions in different models. But our critical question after was to determine whether CLUH loss of function activity alone was sufficient to drive kidney pathology. So to answer to that, we generate an inducible tubular-specific CLUH knockout mice to delete CLUH only in the renal tubular cells. This model allowed us to isolate the CLUH effect of the deficiency of CLUH in vivo. And we observed that these mice developed clear mitochondrial defects with mitochondrial clustering and structural abnormalities, reduced mitochondrial DNA content, impaired complex 1 and 4 activity, and an increased oxidative stress. So metabolically speaking, we observed a shift toward the glycolysis, Warburg-like reprogramming that was interesting too. And finally, structurally, we observed that this kidney of the knockout mice developed interstitial fibrosis, tubular injury markers expression, and a reduced kidney function. So in other words, CLUH loss of function alone was sufficient to reproduce the key aspect of lipocalin-associated pathology that we reported before. So that was a critical validation for us of our mechanistical model here.
Thank you, Morgan. Yes, that was really nice work. Yeah, I'm going to pick up on one thing you mentioned because one of my favorite diseases is ADPKD. So I was particularly excited to see the data you had on that. I think some people in the PKD field are aware that NGAL is elevated in PKD. And a very hot area right now is mitochondrial dysfunction and metabolic reprogramming driving cystic disease. And even being the targets for therapy. So linking this by showing that NGAL is elevated and CLUH downregulated in PKD, in a PKD mouse model, I think was really intriguing to me. And it's buried in your paper, but I hope some of the PKD researchers will pick up on that and take it up. So obviously, all of this is great for mice, but we want to know, does this happen in humans? So what can you tell us about that?
So indeed, one of the aspects for us was to validate our findings in human disease and also assess the clinical relevance. So we decided to analyze CLUH expression in kidney samples from patients with chronic kidney disease. So first, we decided to examine kidney biopsies using immunohistochemistry. In control patients, CLUH showed a strong and diffuse signal in proximal tubular cells, particularly in the basolateral region, indicating that there was a high expression in LC tissues. However, in diabetic kidney disease or in chronic tubulointerstitial nephritis patients, we observed a marked reduction in CLUH staining, especially in the atrophic and damaged tubules. We then extended our observation using transcriptomic datasets from patients with ADPKD, and this analysis showed that CLUH and all its target genes were progressively reduced as the cyst severity increased, while they were preserved in patients with no or minimal cysts. We also found similar results in patients with advanced diabetic nephropathy, where CLUH expression was also significantly decreased. More importantly, we then correlated CLUH expression with clinical parameters, and we observed that lower CLUH levels were associated with reduced glomerular filtration rate and also increased proteinuria, which are two key indicators of kidney dysfunction. At the same time, lipocalin-2 expression showed an inverse correlation with both kidney function and CLUH levels. So as lipocalin-2 was increasing, CLUH is decreasing, and the renal function was worsened. So together, all these findings demonstrate that the lipocalin-2 and CLUH axis we identified in our in vitro and in vivo models were also relevant in human disease. And they suggest that CLUH impairment is also a hallmark of chronic kidney disease progression, and it also may serve as a biomarker of the decline of the kidney function, while it also allowed us to reinforce that lipocalin-2 had a pathological role during the chronic kidney disease onset.
Fabulous. Yeah, that's very satisfying, that it validates what you saw in mice. So do you think this is something that in the pathway that's potentially druggable and useful as a therapy for CKD?
Yeah. In fact, we think that there are several potential therapeutic entry points, actually. The expected one, actually, and we are not the first one to think about that, is that targeting lipocalin itself is probably a good therapeutic entry point here. If chronic lipocalin drives the pathology, we could neutralize its expression in a CKD to reduce the maladaptive signaling. And the approaches that reduce lipocalin expression, such as antisense oligonucleotide, for instance, targeting lipocalin-2 could limit its chronic pathological signaling to CKD. With an emphasis actually on the ASO approach, because we are clearly demonstrating here in previous work too that the effect of lipocalin-2 is the cell autonomous effect by its location in a localization in the cytosol and not secreted form, so which probably is not making an antibody blocking a good approach here. The second therapeutic entry point that we have here is the interaction between lipocalin and CLUH that is particularly attractive here. If we can map, and it will ask more work actually, the structural interface, we could set up different small molecules of peptide that could disrupt this interaction to restore CLUH activity without eliminating lipocalin entirely. And that would be a more precise intervention here. So overall, the therapeutic concept that is emerging from our work, the one of others obviously, is that preserving the mitochondrial homeostasis in renal tubular cells may be a powerful strategy actually to prevent the progression for the injury of chronic fibrosis to chronic fibrosis, and associated with already known and used therapeutic approach could a good way to go actually.
Sure, thank you. Yes. And since NGAL has multiple effects, both the secrete and the intracellular form, are you suggesting that targeting specifically its interaction of CLUH is going to more specifically target the maladaptive chronic effects than the acute adaptation to stress?
Yeah, that's actually one of the approach we are thinking about here, is to have a specific axis on lipocalin CLUH here that will allow us to let the positive or beneficial effect of lipocalin in terms of all these effects on innate immunity response that are really important too, and that can be tuned in this condition.
Well, that's fantastic. And I wish you the best of luck in pursuing that. And hopefully we'll see another paper from you on that very soon. So with that, I think we'll conclude. I want to thank you both, Dr. Gallazzini and Dr. Marques. Thank you for publishing your paper in JASN.
Thanks a lot. Thanks a lot, Alan. Thank you.
This podcast is copyrighted by the American Society of Nephrology. All rights reserved. All content in this podcast is for informational purposes only and is not intended to be medical advice. This podcast should not be used in a medical emergency or for the diagnosis or treatment of any medical condition. Please consult your doctor or other qualified healthcare professional if you have any questions about any medical condition or before taking any drug, changing your diet, or commencing or discontinuing any course of treatment. Thank you for listening to this podcast from the American Society of Nephrology.